everyone, I am Nurul Balqis. In regards of my genomic assignment, I've chosen an article from BMC Genomics, published in 2015. The title is Genome-wide Analysis of LTR Retrotransposon in Oil Palm. What are transposons? Retrotransposons are also called jumping genes. RNA transcribed to DNA by the help of reverse transcriptase. DNA then integrated into the target site of opposite genome. Transposon classified into two general classes, according to its way to changing to another position in the genome. Class 1 is actually autonomous type, and Class 2 is the non-autonomous type. Class 1 has the copy and paste mechanism, and Class 2 has the cut and paste mechanism. This arises in oil palm plantation, which the appearance of mental flower, as shown in the image below, due to the splicing of EGDEF1 gene, which affected the floral phenotype. Also, because of the negative yield that produces abnormal fruits and reduce the oil yield. Four methodologies listed in the article for both oil palm genome. For the first methodology, altered retrotransposon must be in certain parameters. First one, it has to be a repeated element, highly similar 5' 3' ends, and has a target site duplication on each insertion site and has a putative primer and polypyrene track at 5' and 3' ends. Second methodology, gypsy denoted as RLG and copia denoted as RLC. Classification of data are tested according to similarity with domain libraries, indicates many express sequences include functional protein. The third methodology is all retrotransposon with 70% sequence similarity coverage, taken to identify in software. Longer sequence with highest percentage of nucleotide sequence identity are selected. The fourth methodology is the transcriptional analysis of LTR retrotransposon. Quality of data evaluated with FastQC and the quantity of map reads data sequence are calculated. Next is the result of the study. There are 347 consensus sequences of Aedes oleifera comprised of 72% of class 1 and 90% of class 2. And there are 9,915 consensus sequence of Aedes guinensis shows 39% of class 1 and 8% of class 2. In figure 2, both of palm genome has the frequency ratio of Aedes oleifera to Aedes guinensis 5:1. In figure 3, the percentage of identified transmissible element in both oil palm species. In this figure, shows that the full length of LTR retrotransposon is 558 full L-length elements. Copia superfamily displayed the highest rate genome coverage, approximately 2.32%, while Gypsy accounted for 1.06%. This figure shows that shoot epices has the highest expression than other 8 tissue of oil palm that has been tested, and it has the highest level of transcription because of increase of top element and the reduce of that element. Result from figure 6 indicates the estimated insertion of full-length LTR in inner Aedes guinensis genome. There are two peaks of copia elements that have massive insertion, and only a single peak arising for gypsy elements and non-autonomous LTR retrotransposon. Furthermore, the discussion exerted in the article pointed several points. The first point, the distribution of transposable element in the both oil palm are similar. Second, diversity of superfamilies in oil palm genome are comparable between gypsy and copia in the both oil palm genome. Third point is the lineage of retrotransposon comes from retrofit elements and TOC that are active in the oil palm genome. The fourth point is the abundance of copia elements than gypsy elements. It said that copy elements take up 6% of ls genome. Fifth point is the density of retrotransposon across genome. The density across genome shows that copia has preferential insertion whereas gypsy is randomly distributed. Lastly, LTR elements are concentrated in gene-poor regions, and LTR retrotransposon appears to be greater within small plant genome. That's all for me. Thank you.